Welcome to the BioInsights, where science comes alive.
Biological systems are incredibly precise, but the enzymes that run them are not machines with a fixed operational speed. An enzyme does not have a set RPM. Instead, we measure an enzyme's output through its reaction velocity, or V. This is a dynamic tracking of exactly how many substrate molecules an enzyme converts into a finished product within a given unit of time. Watching this velocity fluctuate in real time reveals exactly how external environmental pressures are acting on the enzyme's physical structure. The speed of that chemical conversion is strictly governed by three specific environmental variables: substrate concentration, temperature, and pH. By intentionally shifting these variables and tracking the resulting speed, we can map the exact operational boundaries of any enzyme, whether it is isolated in a test tube or operating inside a living cell. An enzyme's kinetic profile acts as an evolutionary fingerprint. It details the exact physical environment that specific protein was built to survive. We begin with substrate concentration. Imagine an enzyme network with a few scattered substrates. Adding more initially causes a rapid increase in reaction speed, since empty active sites are easily found. But a physical bottleneck eventually occurs when every single active site becomes simultaneously occupied. This state of complete molecular occupation is defined as saturation. At saturation, the system reaches Vmax, or its maximal velocity. Once an enzyme population is operating at Vmax, dumping more substrate into the environment yields absolutely zero increase in the reaction rate. Every enzyme is already working at full capacity. When plotting reaction velocity against substrate concentration, standard Michaelis-Menten enzymes show a hyperbolic curve rising to a rigid plateau at Vmax. Complex allosteric enzymes dictate a different pace. They produce a sigmoidal curve, starting slow, steepening, then reaching that exact same plateau. Whether the path is hyperbolic or sigmoidal, the plateau at the top of these charts represents the strict physical reality of a fully occupied biochemical system. The second major constraint is temperature, which acts as a macroscopic measure of kinetic energy and molecular motion. As heat is applied, molecular motion increases. This causes more frequent and increasingly violent collisions between the enzymes and the substrates. Because of this extra kinetic energy, a much larger proportion of the substrate molecules possess the force required to successfully surmount the activation energy barrier and yield a product. This graph tracks that exact relationship. As temperature rises, we see a steady, linear climb in reaction velocity. This continues until hitting an absolute peak, known as the optimum temperature, representing the perfect balance between high kinetic energy and maximum catalytic efficiency. For the vast majority of human enzymes, this optimum peak lands precisely between 35 and 40 degrees Celsius, perfectly aligning with normal human body temperature. Pushing the environment past this optimum triggers a catastrophic event: thermal denaturation. The excessive kinetic energy begins vibrating the molecule so violently that the delicate internal bonds stabilizing the protein's specific three-dimensional shape tear apart. As the structure unravels, the precise geometry of the active site collapses. Without that exact shape to receive the substrate, the enzyme's catalytic capability is permanently destroyed. This structural failure dictates the precipitous downward crash on the right side of the temperature graph. There are biological exceptions to this limit. Thermophilic bacteria found in hot springs have evolved heavily reinforced protein structures that allow their enzymes to operate safely at extreme temperatures up to 70 degrees Celsius. An enzyme's optimum temperature is the physical threshold where it extracts maximum kinetic energy from its environment, right before shaking itself to pieces. The final physical constraint is pH, which controls the overall concentration of protons in the enzyme's immediate environment. Catalysis relies on highly specific functional groups within the active site being in a strictly defined ionization state, like a protonated amino group snapping cleanly onto a substrate. Shifting environmental pH strips these critical protons away, instantly disabling the active site's ability to bind. Furthermore, extreme shifts in pH will also sever the internal ionic interactions holding the protein together, causing full structural denaturation. Because the pH environment varies drastically across different parts of the human body, enzymes are custom-tuned to operate strictly within their local biological zones. Most enzymes found operating in the cytoplasm or the bloodstream are calibrated for a near-neutral physiological pH of around 7, but the digestive tract forces enzymes to operate as extreme outliers. Pepsin achieves its maximum reaction velocity in the highly acidic environment of the stomach at pH 2. Most other enzymes would immediately dissolve here. Just a few inches away from the stomach, the environment of the small intestine requires a completely different approach. Here, the pancreatic protease trypsin operates. It reaches peak velocity in a slightly alkaline environment of around pH 8, driven by the presence of bicarbonate. These specific pH peaks demonstrate that enzymes are specialized catalysts, optimized for highly localized chemical environments. Achieving a high reaction velocity requires a demanding set of physical conditions. The enzyme needs an abundance of available substrate, the exact kinetic energy to drive the reaction forward, and the precise electrical charge to hold its structure intact. A severe deviation in any single one of these three environmental metrics is enough to shut down an entire metabolic pathway. These laboratory curves represent physical constraints. They define the exact physiological tolerances a protein requires to remain structurally intact. These requirements are hard-coded into the protein's sequence over generations, a record of the specific conditions it was required to navigate. By mathematically analyzing where an enzyme fails, we reveal its lineage of environmental adaptation, showing exactly which corner of the natural world it was built to dominate.